I guess we're just going off schedule. All right. I'm Felix Beaudry. I'm from the University of Toronto. I'm going to talk today about selection interference and haploid purging on Y chromosomes in Rumex. What I'm really interested in, we've heard a lot about changes in recombination, and we just heard a wonderful story about Y chromosome degeneration, and that's the process that I'm going to talk about today. This process where we think the X and the Y used to be the same size, and now we look at them and we see that the Y is often a very different size than the X. And what we think is happening here is this process of selection interference. So if we think about two X's, which are recombining in females, and we take one of these horizontal lines as a genome and one of these little skulls as a deleterious mutation, and these little dotted lines of recombination are like crossover locations, we can shuffle around the deleterious mutations, and then if we remove the most loaded haplotypes, so the ones here with two skulls, there's still lots of genetic variation to play with. But if we're on a Y and there's no recombination, and then you remove these most loaded backgrounds, you can actually cause the fixation of these slightly deleterious mutations. And over time, that's why we think we end up with this tiny little Y as we see in humans. But in plants, it might be a little bit different. And the reason we might expect that is this alternation of generations. So this is, for those who are fuzzy on alternation of generations, this is the idea that the flowery, kind of leafy part of a flowering plant is diploid, but a large portion of the life cycle of a flowering plant is haploid. So this is the pollen or the ovule. And why this might be important is that in a haploid tissue, anything that's expressed is unmasked. So if you have a recessive allele and it's expressed in a haploid tissue, its effects should be felt. And this can be really important for sex chromosome degeneration. So if we think a new deleterious allele is recessive and it's expressed in leaf tissue, it can be masked and allowed to spread and fix in the population. But if it's being expressed in pollen, this deleterious allele on the Y can actually get removed from the population, and degeneration on the Y can get slowed. And so to give you an idea of the scope of this, if we, in different plants that we've estimated, or that different groups have estimated the amount of the genome that's being expressed in pollen, it's about a bit less than 50%. If we compare that to animals, about 5% of the genome is being expressed in sperm. And so we should have this interesting balance between haploid purging, so the removal of these deleterious mutations on the Y, and this selection interference, this buildup of deleterious mutations on the Y in plants. And so the question that I wanted to ask today is, do we see signatures of haploid purging? Is this a real thing? Are pollen-expressed genes on the Y less likely to be deleted? So that would be this sort of balance between degeneration and haploid purging. And then I'll talk about whether we can use genetic diversity on the Y to predict the sex ratio bias a little bit later. So I study this question in Rumex statulis, which is this plant, this dioecious wind-pollinated annual. This is the Y chromosome of Rumex statulis. It doesn't look as small as the human one, but we've done some work showing that there is indeed molecular degeneration and the fixation of deleterious mutations. And so what we wanted to ask is, do we see these effects of haploid purging? And to do that, we did some RNA-seq of pollen, and then we compare that to RNA-seq from leaf tissue, and then we use DSeq to look at whether there are genes that have more reads from pollen compared to leaf, and call those leaf bias, and vice versa. More reads in leaf than in pollen are called leaf bias. And so the first question I want to ask, can we see the signatures of haploid purging? And one of the cool things we did with this data set is our RNA-seq for the leaf was taken from all over the range of this population. So we have allele frequencies in natural populations for both our leaf and pollen-biased genes. And what we can do is look at both synonymous and non-synonymous sites and see the frequency of alleles in either of these biased tissue genes. And we can look at how many synonymous mutations are segregating in the population and compare that to non-synonymous to get an estimate of how often non-synonymous mutations are getting removed by purifying selection. And with those data and using DFE alpha, we can estimate the fraction of sites that are under different levels of constraint. So on the left, we have the most neutral, the fraction of sites that are evolving approximately neutrally, and as you go to the right, you'll see higher and higher constraint, and then we can compare that for leaf and pollen. And we see that in our highest constraint, there is an overrepresentation of pollen-biased genes. And although this isn't independent because it's actually a fraction of site, we see the converse to be true, where in the lowest neutral bin, we see an overrepresentation of leaf-biased genes. And so we do have evidence that this haploid purging is happening. So the next thing we wanted to ask about is, are pollen-expressed Y genes less likely to be deleted, as we'd expect from this pollen purging? So what we did here, this is work with George Sandler. What we did here is we took hemizygous genes, so these are genes that have lost their Y copy, but the X copy has been retained. And we looked at the proportion of those genes that are leaf biased versus pollen biased. We compare that to genes that have retained their Y copy. So there's a copy on the X and a copy on the Y, and then compare that to the autosomes for like a genome-wide standard. And what we see is that genes that have been lost from the Y, so this hemizygous category, is way overrepresented for leaf-biased genes, and genes that have been retained on the Y are more likely to be pollen-biased. And so we do see this evidence that pollen-expressed Y genes are less likely to be deleted. And so the third part of this talk is something I think is really neat. And so I wanted to see if we could use genetic diversity on Y chromosomes to predict the sex ratio bias. And so what's happening here is, if we take this haploid purging to a population, what we're saying is we're removing these Y chromosomes disproportionately compared to X chromosomes, which should mean if these pollen grains are competing to get to ovules, we might actually see that the next generation is female biased because we have less Y chromosomes in the population. And so I wanted to ask, can we use the signals of these backgrounds that are getting removed to actually predict the, and in neutral genetic diversity, to predict the female proportion in the population? And so how this would look is, if we increase the number of linked sites under selection on the Y, we should imagine that the effect of this interference selection is getting more serious, so that we have both a more sex-biased ratio in the next generation as we're removing more Ys, and we're also removing more genetic diversity. So what I did is some simulations of this process in SLIM. So as we go towards the top right, we're increasing the number of linked sites under selection. And so here we use a non-Fisher-Wright model, which means we can remove the least fit individuals. I've chosen a selection coefficient of minus 0.03, which is the estimate we got from our DFE alpha, which I was showing earlier. We do this 100 times. The 95% confidence intervals are there. They're just right behind the dots. They're tiny. And we do that 100 times, and we wait to get to equilibrium. And so what I think is really cool is this does show that there is potential to get this, to get an idea of the amount of genetic diversity being removed and the sex ratio that we see in the population. And we're seeing that there's this first phase, this background selection phase, where only neutral alleles are getting removed. But as you add more and more linked sites, we actually get to the point where deleterious mutations are affecting each other's fixation, and that's where you start getting these deleterious Ys that are getting removed from the population. Now what we can do is actually take our empirical estimate of the sex ratio bias in Rumex of Statulis by Melinda Pickup and Spencer Barrett. And what we estimate is, or what they estimated was about 60% female biased. And then we can look at the genetic diversity that we see on the Y compared to the autosome. As you can see, my simulations don't match what we'd hope the intercept would be, so I'm still revising this simulation. Hopefully we can find an explanation for why the genetic diversity on the Y is lower than we would expect from the sex ratio bias. I'm thinking about adding beneficial mutations and adding a more realistic demographic model. But this is where this part of the project has gotten. So overall, we do see that haploid purging is having an effect on the genetic diversity that we see in the genome. We see that genes that are expressed in pollen that are on the Y are less likely to be lost. And unclear, maybe we might be able to use the genetic diversity to predict the sex ratio bias. And with that, I'd like to thank the University of Toronto, the conference for hosting me, the Wright Lab with a specific shout out to Team Rumex, and a bunch of collaborators. And I will take any questions. I don't really know how much time I have. I don't know that there's, or who's next? Great. Okay, well, I'll pass it off and thanks for